Hi everyone, we are from Group 9. We're presenting about the growth factor. For this one, I am Nura Ain Shafika binti Muhammad Khori. As a presenter, we present on EGF and ErbB. Okay, EGF, or the full name is epidermal growth factor, is a molecule of protein that it will act as a primary messenger that later will initiate the EGF signal transduction pathway. Next, EGF has receptor that consists of two identical but inseparated monomeric units, and it has two types of monomer, which the first one is extracellular site and the second one is intracellular site. Extracellular is the binding site for EGF, while the intracellular contain tyrosine kinase. And the roles of the EGF is stimulate the growth of the epidermal and the epithelial cell by binding to its receptor, which is ErbB1. There are 6 important protein factors, which is GRB2, SOS, RAS, RAF, MEK, and also MAPK. Okay. For the pathway, EGF binds to the epidermal growth factor receptor EGFR on the cell surface, and then the ligand will induce dimerization. So the conformational change of extracellular receptor will change the protein tyrosine kinase, and it will activate the protein tyrosine kinase, and as well as it will later initiate a signal transduction cascade, and it will resulting in a variety of biochemical changes within the cell, such as it will increase glycolysis and protein synthesis. Second, it will increase in the expression of certain genes that later will lead to the DNA synthesis and the cell proliferation. Next, for the ErbB. ErbB is a protein receptor for epidermal growth factor to bind. The ErbB family of protein contains of 4 receptor tyrosine kinase, which is ErbB1, ErbB2, ErbB3, and also ErbB4, that is structurally related to the epidermal growth factor EGFR. ErbB1 overexpression is at lung cancer, head and neck, and also brain tumors. ErbB1 interval deletion is in the glioblastoma multiforme, and ErbB1/2 kinase mutation is NSCLC. ErbB2 amplification or overexpression is at breast, ovary, lung, uterine, and also stomach cancer, while for the ErbB4 mutation is at melanoma. Okay, here is a picture of the ErbB receptor, which is active as a dimer. EGF protein will only bind to the ErbB1 because it is specific. Okay, last but not least, I will present on the EGF mechanism involved ErbB1. Okay, here is the picture of the mechanism, EGF mechanism. Okay, when the EGF signal molecule binds to the ErbB1, then it will induce the dimerization, which then lead to dimer formation. This is the dimer formation. The conformational change then occur, followed by cross the phosphorylation, where the carboxyl terminal end of one tyrosine kinase domain move into the active site of the opposite tyrosine kinase domain. This phosphorylate the C-terminal tail and create attachment point for the other protein. And after that, GRB2 anchor to the ErbB1 phosphorylated region, and then GRB2 will bind to the SOS followed by the SDS binding to RAS. Okay, then we continue. The RAS expel GDP and binding a GTP. The activated RAF then move to activate the protein kinase MEK. The activated RAS then move to activate the protein kinase RAF MEK and then activate the extracellular signal regulated kinases ERK. The activated ERK move to the nucleus and stimulate the transcription factor to increase the gene expression, which is to increase the rate of protein synthesis and lead to the cell growth. Next, Fazlin Hassan will present on the next growth factor.